So our next speaker is Professor Zvonimir Dogic from University of California, Santa Barbara. He's going to tell us about active membranes and foams.
Okay. Thank you very much. It's very nice to be here, especially since this is a celebration of Sriram's contribution to science. It was a 22-hour flight, which is a pretty strong repulsive barrier, but, being here and Sriram's contribution was strong enough to overcome this barrier. And so Sriram had, important influence on my scientific career and what I do. And, this is perhaps one of his better known papers in two decades ago, where he showed that, if you have extensile rods, that they will exhibit this ubiquitous bend instability. And you have contractile rods, that they will, exhibit a different kind of splay instability. And this came just about the time when I obtained my PhD, and doing PhD, I had a great time, and I just wanted to continue doing what I was doing, which was self-assembly of colloidal rods. And people in the community said, well, maybe you should think about branching into new areas. And it took a few years to get that message, and I didn't know what to do exactly, but this kind of theories that were being developed were certainly something that, as an experimentalist, gave you a confidence that once you jump off the diving board, that there's going to be some water in the swimming pool. And luckily, Sriram turned out to be correct, and so there were experiments to be done in this field. And so what I want to talk to you about today, and this was, I think it's, a lot of, especially my own lab has focused on this unique bend instability that, we've been studying for a number of years. But what I want to talk about today is what happens when you tune the system so that it lives at a boundary between this contractile and extensile system, because then you can imagine that the active stresses reorganize your rods, the rods feed back and give rise to new kind of mesoscopic active stresses. And as you will see, the dynamics that emerges in this regime is quite complicated. So if there's any questions during my talk, if there's about the geometries that I'm showing you, please stop and interrupt. So this is something we've been working on for no? Okay, this should work. This is a aligned rods, extensile rods, and they undergo this unique bend instability that, Sriram has pioneered. And so these rods are essentially microtubules, and you have this kinesin molecular motors that bind anywhere along the microtubule length and push the microtubules apart, generating these extensile stresses that, I was, that Sriram pioneered, or at least that's what I taught for a while. And then I was recently talking to people who are much more broader in their world art view, and they pointed out to me that bend instability was actually discovered before Sriram's 20, he got scooped by a few years by Saul Levitt. Here you can really see this abstract art, this bend instability, and it has certain appeal. And I must say that, he made quite, how shall I say that, financial profit by inventing this particular bend instability. Okay, so I told you before that, what we've been looking for is molecular motors that bind anywhere along the microtubule and induce extensile motions. Now, I think everybody here would agree that, we really don't know yet how changing microscopic motor dynamics influences microtubule self-organization. And so we've put Okay, I have to go back and forth. So we've started working with a new kinesin 4 motor. This is a kinesin motor that is imported in a mitotic spindle. This motor lands along the microtubule, walks along the microtubule, comes off, but once it reaches an end of a microtubule, it tends to linger there for much longer than kinesin 1 molecular motor. Kinesin 1 molecular motor does this as well, but kinesin 4 to a much greater extent. And so this is a single molecule experiments from the lab of Radhika Subramanian, where she labels kinesin motors, and you can see that they start assembling this cap. And depending on the biochemical conditions, more or less a tiny fraction to almost an entire microtubule can be decorated with this kinesin 4 molecular motors. And if you're a soft matter physicist, what this might remind you is something like a block of polymer, right? Because now I have a rod that has one end that has kinesin motors, and these kinesin motors tend to be very sticky, and then you have a naked microtubule end over here. And so this is certainly interesting in itself, how this spatially heterogeneous filament structure is encoded not in the chemistry like in a diblock of polymer, but in microscopic motor dynamics. But what we want to focus on, we want to take something like this and then ask the question of what happens when you put many filaments together in a suspension. And so what you'll see initially is that this system is going to be contractile initially. And so you can see over here, you start observing dense formation of microtubules that increase in density as time goes on. And in the end, you can see that there's almost aster-like structures. This is certainly not so surprising. People have seen that in many other systems before. And so the idea here is that you have a motor, the microtubule has polarity, the motor walks and concentrates all of the microtubule plus ends. And what happens here is you have a rich, motor-rich core and radially protruding microtubules pointing outwards, right? Now what you can do is you can play around with the conditions a little bit, and so that initially your contractile instability actually leads to larger structures, right? And so this is now still anisotropic, and I can discuss that later, a contractile structure that it's much bigger than what's been observed previously. And what you see over here is formation of a radially outward microtubules, but now you have an interior of microtubules, of slightly enriched microtubules, but this almost looks like a vesicle-like structure. As this instability develops over time, yes, this is probably 10 microns, 15 microns in size. This layer of microtubules is about 10 microns in thickness, right? What you can observe is that this contractile, this layer that forms at the boundary of the contractile structure is usually fairly uniform in size. But what happens over time is that it starts breaking up, and there's a lateral transfer of microtubules. And you can see that at this point, all of the microtubules actually accumulate at the sharpest edges of the structure, right? And so I believe that this is certainly possible, that this is an example of a Shriram-like instability, but one for a slightly different organization of microtubules. By the way, it says that I have infinite time here, so I can just keep talking. Zero, or I'm already out of time. It's flashing red, so that means I should just stop. Huh? Okay, so let's go on. So this was certainly surprising. You have these kinesin asters that are contracted, and one particular time, the system completely switches, and at this point, the kinesin asters start expanding. So there's a very sharp transition, and you can see that over here, right? As again, this movie will play loop over. So we started with a maximally contractile structure, and then, you know, a certain number into the experiment, the contractile stresses switches automatically, and these asters start expanding in time, and you can see that this layer of microtubules at the edges becomes more and more dilute. So maybe I'll wait for this movie to play. I'll play it one more time, because this is a important summary of what happens, right? Is you have these asters, and over time, these asters, which were contracting until that point, start expanding, right? And eventually, they almost overtake the entire sample, right? Now, if you have these expanding asters, at some point, a pair of asters will simply encounter one another. And this is, so what we can do is we can quantify this dynamics. Yes? No, this is a three-dimensional, this is a two-dimensional cut of a three-dimensional sample, right? So Remy Boros, who's been working on this, has actually done a really good job of quantifying the size of these, quantifying where these expanding asters are located. And so let me just show that over here. The image analysis reveals these, you again have this maximal contraction, and over time this maximal contraction switches to expansion where these asters are accumulating material and grow with time. Yes? You'll come to the, yes, that's a good point. You'll see what happens at the end, right? And so what you can see here is you have density distribution, and you can see that the density distribution grows, you reach maximal contraction, and at some point, the contraction stresses switches to extensile stresses, and you get expansion of these droplets, and they expand and encounter one another, right? And so their density decreases, and their volume of these vesicles decreases and reaches a steady state. So what happens is you can also visualize what is the map profile across the vesicle edge. This is a vesicle edge. This is the outside of the vesicle. This is the inside of the vesicle. You get this development of a very well-defined radial peak, and this is of the order of 10 microns, which is more or less consistent with the microtubule length, contour length. So this is a monolayer or a monolayer-like structure of microtubules. Now what you'll see is these vesicles, these monolayer vesicles are expanding, as you can show in this movie, right? Is they're growing with one another, and they'll encounter one another, and they'll spontaneously coalesce, as is shown in this movie, right? So there's a boundary here, and very quickly this boundary is completely annihilated, and you get a larger droplet vesicle, right? And so what we believe is happening here is, again, you can see that in this movie, is the vesicles come together, they annihilate, the microtubules between the vesicles completely disappear, and the vesicles grow and become larger, and they're very anisotropic, as is shown over here. So what we believe is happening here is that you have these monolayers, you have microtubules over here, the motors are shown in blue at the base of these layers, and what can happen is that the microtubule protrudes. There's a lot of motors in the background suspension. The motor can cross-link two microtubules, and then what's going to happen is this is going to increase the force, and that's going to simply lead to these monolayers interpenetrating one another, and we believe disassembling in ways we don't understand. But as soon as you establish a connection, there's more sites where the motors can be recruited, and this thing very quickly self-annihilates and falls apart. What we can do is we can also measure, okay, do you have a question? Okay, please.
Yeah, no, I was trying to understand that. So it's a monolayer, but
Yes.
the polarity of the microtubule is kind
polar.
It's a polar monolayer. Each layer is. So it's,
Yes.
it's a little different from a vesicle in the sense that normally both sides
Yes.
of the vesicle would
Yeah,
be the same.
this is a monolayer.
I see.
This is a
So
vesicle-like structure with a polar.
So it's like it's like a microemulsion droplet or something.
Yes,
meaning
maybe.
The inside and the outside aren't quite the same
yes,
because of the
yes,
polarity to the
yes,
normal.
Yes, this is a polar structure, right? And you can see that the blue is the motors, and you can see the motors at the base of the vesicle.
So what's inside the vesicle? The same thing as outside, or is it
it's empty space, a little bit higher microtubule concentration. There's still microtubules here and here, and they're always going into the membrane and out of the
Are
membrane.
there free motors inside the vesicle or not?
The motors are there's a large concentration of the motors in the background, and then they come to the vesicle, to the microtubule, and bound to a microtubule and fall off. So they are all continuously in a dynamical steady state. So this is just another movie. Here we've actually labeled all of the membranes with the very dense red microtubules, and then we visualize individual microtubules. And here you can really see this very rich dynamical pathway. So again, let's the movie come to the beginning, and so you'll see the initial instability. Oh, let me play that one more time. You get this initial instability, the contraction, and then the contraction switches to expansion, and these vesicles start coalescing with one another, and you get this very free-flowing, highly active dynamics of these microtubule monolayer sheets that in principle should be polar. And so those are the initial stages of the phase separation. What you observe is isotropic contraction that leads to the formation of maximally contracted asters. At certain point, these asters expand into monolayer vesicles that continue to expand and coalesce with one another, and then you end up with this kind of layers that are continuously being reconfigured by active stresses. If there is no activity, the sample is completely dead and inactive. If you reduce the microtubule concentration, what you'll see is you'll essentially end up with an isolated single vesicle, right? So here you can see the vesicle is contracting. This is an interior of the vesicle. The microtubules are pointing over here. This is the layer enriched in the motors. You can see this initial instability of the motor densities. The motor densities smoothen out over time, and the vesicle membrane grows. And now you can see that the membrane starts deforming, right? Because presumably there's some active stresses. They could have different symmetries. I think this will speed up with time a little bit, and you'll see that this this very large monolayer starts deforming and bending. These were the experiments done at very low kinesin concentration, so the activity is very slow. The other thing that I should say is that previously we've looked at everything in XY plane. This is a view in XZ plane, and so this is now a cross-section of essentially a very large vesicle. The ends are somewhere over here, the end is somewhere over here, and this vesicle is exhibiting fluctuations presumably driven by active stresses. And again, this is the idea. You have this monolayer microtubules, and you have the base that's enriched in molecular motors. If you increase the concentration of the molecular motors, you observe very much similar dynamics, but now the deformations of the layer are significantly more pronounced. Again, those are the initial instabilities that I've described previously. And what you end up with is, again, you end up with this surface layer. Now the surface layer becomes, because there's many more motors, becomes much, much more dynamic, right? And you can see that these protrusions are growing, but they are only growing inward into the vesicle and not outside the vesicle, right? And so, for example, over here, you'll have a protrusion, and you'll see that it completely disassembles very quickly as this dynamics is taking place. So what do we believe is happening here, right? Is you get an active stress, it drives curvature in one direction. Presumably that's okay because this is a polar structure. You have an arrangement like this, and then you have a neck here, and then this neck is pinched off, and then you have a vesicle that simply self-annihilates. So what we believe is happening is you essentially have over here, because this is the arrangement of the motors, right? And now this motor's very similar to the vesicle annihilation effect when you have these bending fluctuations, are pointing towards one another, and whenever the microtubule ends encounter one another, this becomes unstable and it pinches off. And then you have a vesicle, but this vesicle is, has motors on the outside, and there's motors on the inside, and as soon as the motors come on the inside, they will push the microtubules together, and this thing will very quickly disappear and evaporate, and that means that, what you have is you have active stresses that generate curvature, and this curvature leads to these very large invaginations, which leads to unstable configuration, which leads to microtubule, the monolayer disassembly. that presumably produces new material that can be next incorporated into this membrane structure. This is a three-dimensional movie of one of these two surfaces, and you can really see these protrusions. They very quickly disassemble. They grow in one direction, that means upward, and as soon as you reach a critical concentration curvature, they very quickly disassemble, and so you have this very unique dynamical state. Okay, so this is the intermediate states, right? On the very long time scales, what you observe is something like this. Again, you've seen this dynamics many times again, like you have this aster formation, you have this expansion of the asters, and you'll see now there is this merging events, right? And so curiously, these droplets are very, very anisotropic. And what happens is at some point, the two bases will reach one another. And at this point, you no longer have a polar bilayer, but you transform into something that is an apolar two microtubules on each side bilayer, right? And you can see this dynamics shown over here, for example. Again, you have these vesicles driven by active stresses. They're very aspherical, and you can see the zippering action over here, and then you end up over here with a bilayer instead of apolar bilayer instead of a polar monolayer. Putting all of this thing together, now I've changed the color scheme. It was too late to modify this movie last night. Again, this is the dynamics. You have these apolar bilayers. They start condensing now into a structure that really reminds oneself of an active foam, right? So you can see that these bilayers are exhibiting very rich dynamical behavior. They're moving, and so this is a three-dimensional foam-like structure exhibiting interesting dynamics. Again, this is just a different movie. Now, this is not in time. This is a Z projection, and you can really see if you follow one of these that this is a bunch of closed cells, right? And so you end up with something that is presumably what an active smectic might look like, right? It's disordered foam-like material with these cells that can be either open or closed, right? Now, what's interesting is if you look at this foam, you'll see these flashes, right? And these flashes are very characteristic. And then at this point, actually, this three-dimensional foam dies, and you can see a slight expansion when the motor activity is set apart. And you can see these flashes, and what these flashes are, it's essentially an individual cell reaches a small enough size where the two bilayers collapse onto one another, and an entire cell can disassemble and provides new material that builds wall somewhere else. So that's essentially the end of my story. So let me just give 2 minutes of conclusions. You have this long-term steady state. You have these active foams. There's a number of interesting observations. You have contractile dynamics that switches to extensile dynamics. The molecular reason for this switch is not understood for us. We have some ideas. You have these active polar solid membranes where you have this non-equilibrium bending fluctuations that can couple with the local disassembly when you reach these critical curvatures. And again, you have these active apolar forms that exhibit this very unique non-equilibrium pulsatile dynamics. Now you might ask, is this just some weird thing that we've seen in a particular system? And we've seen some evidence of similar like behavior, right? So for example, here we are changing actin concentration. You have extensile dynamics, you have contractile dynamics. At a transition point, again, you see these aster-like structures where the core of the aster is enriched in actin and there is a microtubule layer around it. And these active asters exhibit very similar like merging dynamics. So here again, you have this merging of two actin cores, and then the microtubules open up, the layers disassemble as they counter one another, and you bring the structure together, and so that's a very similar motif, right? And finally, just as a last minute, there's very interesting microtubule dynamics in xenopus extract eggs. So for example, under certain conditions, you can get these microtubule asters, and over time, you'll see that these microtubule asters open up and form a cell-like structure. And in a certain way, this is reminiscent to what we are seeing. It doesn't mean this is a much more complicated system, but interestingly enough, this kind of contractile asters can actually spontaneously start dividing. And I believe that there's actually a lot of active matter physics associated with this. So with that, I would thank you for your attention and for all the questions that you've already asked.
When the vesicles are coalescing, is there any rule of surface tension, or is it entirely activity driven?
No, these vesicles are nothing but spherical, right? There is active stresses that drive these very large deformations of these vesicles, right? And we would very much like to understand what is the structure of these vesicles, what determines these non-equilibrium fluctuations of these vesicles, and what is the origin of the active stresses, because you can imagine that, they could be Sriram-like contractile instability, but there could also be polar stresses due to the polar arrangement.
So in the actin microtubule composite that you showed,
Yes.
you showed the merger. Do you also
Yes.
see like a cell
Yes.
division thing?
Yes.
And I didn't get the concept. Actin is on the periphery, or is it microtubule?
No, the actin is encapsulated, and microtubules form this monolayer that is encapsulating actin. And what I should say is that this is an entirely different system, entirely different motors. Actin, the only thing is that similar is microtubules. But again, by tuning the system between extensile and contractile instability, you observe very interesting dynamics where there seems to be a feedback where active stresses drive the microtubule organization, that they can switch the active stresses or the symmetries.
So why do the microtubule asters divide? It's just the expansion that forces them to divide?
What drives the expansion?
What drives the expansion?
I mean, again, like we are doing quantitative imaging. You can imagine many possibilities, right? They are highways, and what can happen is that the motors are continuously moving along the microtubules and coming to the core, and then the core has to open up because you can't build the infinite density, right?
But you have a continuous flux of motors because all the microtubules
Yes.
are oriented. How do you feed them back inside the structure?
I don't know, and there's also microtubules being continuously pulled in. One possibility is that initially when the system that's the experiments we are doing right now, we don't have quantification of what the motors are doing into initial stages, right? So what you can imagine is that initially the microtubules are apolar in an aster, and then they polarity sort, and that induces the transition in the behavior, but that's a hypothesis only.
Is there time for one?
Last question.
Okay. What you call the active foam, it's basically bilayers dividing two sides that are more or less the same.
Yes.
So
yes.
it's a bit like some weird active version of what people in the surfactant in solution literature would call the L3 phase, the sponge phase, which is just, it's fluid random surfaces on scales much bigger than their persistence length.
Yes.
And basically what you'll have then in those systems is, of course, there you can change the concentration, and you have a phase transition in the equilibrium case, in the Ising universality class, where all you can measure is the energy-energy correlator, because that's the thing that marks the interface. So this could be an active sponge phase. It could be, right? I mean,
under different conditions, you can obtain entirely closed structures that are topologically separated. But if you change microtubule concentration, there will be openings,
right?
And there will be an open kind of.
Right. But I guess there are also free edges, or there are no free edges?
There is free edges. Unlike foam, these things can grow a bilayer that will grow over time. And so I mean, certainly it's not a strict analogy with the foam.
We'll have the next speaker, Professor Chandan Dasgupta.
Thank you.